Hello and welcome. My name is Dylan Lauki. My metric number is 206480. In this video, I'm going to demonstrate the preparation of bacterial smears first and then gram staining. These are the materials you needed for bacterial smears. These are the materials you needed for gram staining. First of all, flame your inoculating loop until red hot to completely sterilize it. Then, cool it down for 10 to 15 seconds. After cooling down the loop, dip the loop into a tube containing sterile water and place it on the slide. The slide should contain one loopful of sterile water on it. Then, touch the tip of the loop on a solid medium containing culture and then transfer it onto the slide. This, you will create a suspension. Spread the suspension in a circular manner until it is about the size of a dime. Remember to label your slide. Then, flame the loop again to completely sterilize it after using. After you have successfully created a bacterial smear, allow it to air dry completely. After the bacterial smear is completely dry, heat fix it over a Bunsen burner. The heat fix process is important to allow the bacteria to adhere completely on the slide. If you are transferring bacterial culture from broad culture instead of solid medium, you do not have to use sterile water. Instead, first of all, flame the inoculating loop until it is red hot to completely sterilize it. Allow it to cool down for around 10 to 15 seconds. After that, flame the neck and mouth of the tube and dip the loop into the growth culture. Flame it again, close the cap, and then smear your bacterial culture onto the slide. The bacterial smear should be about the size of a dime. After smearing, flame the inoculating loop to completely sterilize it after use. Then allow your bacterial smear to completely air dry. After it has air dried, heat fix the bacterial smear to allow the bacteria to completely adhere to the slide. And with this, you have successfully prepared a bacterial smear.
This is the example of a good bacterial smear. A good bacterial smear should not be too thick or too thin to allow clear observations on the microorganisms.
After preparing your bacterial smear, then you can start with your gram staining. First of all, start with flooding crystal violet on your bacterial smear. Then let it sit for 1 minute. After 1 minute, then gently wash the stain with tap water. Remember to not run the water directly onto the bacterial smear. After washing with tap water, flood the bacterial smear with a mordant, which is gram iodine. Let it sit for 1 minute. After 1 minute, gently wash the slide with tap water again. After washing the slide with tap water, then use 95% ethyl alcohol and run it drop by drop onto the slide. Apply it gently until the alcohol runs almost clear. Caution should be taken so that the alcohol do not decolorize the stain for too long or too short. After applying alcohol, gently wash with tap water again. Next, gently counterstain the slide with safranin. Let it sit for 45 seconds. After 45 seconds, you can wash off the stain with water. Then, blot dry the slides using bibulous paper gently. After blotting dry the slides, you can observe it under microscope using oil immersion.